The work that they do themselves screams out that life did not start this way. Because even with all of their input, even with all of their human-made reagents that are only available in a modern laboratory, plus they had to start with all of the basic components came from living systems. Have scientists demonstrated that RNA could replicate itself? That question has arisen many times, and that's what we're going to address today. So can these molecules really start moving, or as some people would claim, evolving toward life? And that's what we're going to look at today. And I have my regular guest, Rob Stadler. And just so that you know something about Rob, he got his undergraduate degree from Case Western Reserve University. And then he got his PhD from Harvard and MIT. It's a dual program in the Division of Health Sciences and Technology. So he has a very strong background in these sort of things. And Rob's going to share with us today and give us his thoughts on this topic. So Rob, welcome, and take it from here.
Thank you, Jim. It's always a pleasure to talk about RNA replicating itself. I'm sure that gets everybody's pulse up. So we're going to talk about today this topic because there's been a lot of recent publications on this, and it's nice to review those publications and see where the science is at the current moment. So as Jim mentioned, the big ultimate goal, the audacious goal here for these researchers is to try to find a totally natural explanation for the origin of life. Just letting physics and chemistry do its thing. How did life get started? That's a big challenging goal, and there are many, many difficulties. But one that we're going to focus on here is that life requires these very complex biopolymers like DNA, RNA, proteins, and polysaccharides. And if you ask anyone who knows probability, they'll tell you that the kind of complexity required to start life is impossible, really, for random processes to generate. Even if you have the entire 14 billion years of history and the mass of the entire universe, there's not enough probabilistic resources, not enough opportunities to try to generate these kind of codes, these information-containing molecules. So the remaining hope for people that are looking for a natural explanation for the origin of life is this process called chemical evolution. And that involves these 4 steps shown here. First, you have to have self-replicating molecules, and that's our focus for today. And they're not just self-replicating molecules, but they're information-containing molecules that have to copy and pass that information along to the next generation. And that replication process needs to have only a few errors occasionally. If you have too many errors, you end up with what's called error catastrophe, meaning that the information you have is lost, it's dissipated, and you're back to a random state again. And then you need to have a strong natural selection filter to prefer the more fit molecules and get rid of the molecules that aren't fit. And then it's envisioned through that process that you have a gain of complexity and function over time. So you have a molecule that can replicate itself, and it becomes better at it, and that takes over the population of molecules around through that natural selection. And then it starts to pick up other functions, like maybe it can catalyze some metabolic reaction or try to contribute to homeostasis or so forth, all these things building up toward life. That's the vision. But for chemical evolution like this to contribute to the origin of life, this kind of self-replication must be quite pervasive. It's not just a single molecule that can do it, but it has to advance to another molecule that can do it, and another one, kind of like steps in a ladder, each one of those steps having self-replicating molecules. And then other functionality has to develop, like I said, metabolics or homeostasis, and those also have to be self-replicating. So a whole army of self-replication going on, moving towards life is the vision. But this has to be quite pervasive in order to be successful. So you would think, if it's that pervasive, that it wouldn't take us 50 years of research to try to find one. But that's the state we're in. And as Jim mentioned, RNA is the most likely molecule, the so-called RNA world hypothesis. And that's because it can store information and it can also have enzymatic properties called ribozymes. They have some catalytic properties inside the RNA. So from this big, audacious goal of finding the explanation for the origin of life, we're going to chop it down to a small sub-goal, which is just to demonstrate that RNA can self-replicate, to show that this chemical evolution concept at least has some hope to it. And as Jim mentioned, a lot of people have already claimed, a lot of hype, a lot of media hype, a lot of YouTube hype, that self-replication of RNA has already been shown. And I wanted to make it clear, this is not me saying it here, but this is from one of the papers we're about to review. And these are authors who are striving for this goal, and they say quite clearly, the ability for RNA self-replication has not been observed in nature. Okay, so you don't have to take our word for it. Take the experts who are actually trying to achieve this. So I wanted to take a little moment just to talk about how unnatural RNA self-replication would be. Phil Hollinger, one of the investigators on this paper that we're going to talk about, he gave a talk this week at the Molecular Origin of Life conference. And he actually said it's rather perverse to think that RNA can self-replicate. And what he means by that is that there's a lot that we know about natural laws that goes against the concept of RNA replicating itself. I mean, we know enough about physics and chemistry to know that all of the forces of nature gravitate against this concept. And another analogy for that is something like a perpetual motion machine. You know, and here in this cartoon, it says for years he had spent every free moment working on his perpetual motion machine, and so far he was up to 18 seconds. And I think some of these origin of life researchers might feel a little bit like this. Now, I know it's not exactly the same because perpetual motion is something that people used to work on, and now we know enough about physics and thermodynamics to say it is impossible, and that is clearly a closed form solid answer. When it comes to self-replicating of RNA, I can say that natural processes oppose it, but we don't yet have the complete understanding in closed form to say that self-replication is absolutely impossible, but it sure is not likely to happen from what we know about how natural processes work. If you're interested in a little more detail on that topic, I would recommend this long story short video called Debunking RNA World. This is something that I had contributed to, and it's enjoyable to watch. It gives you some background. But if you want the short version of that, I will tell you just eight little facts here to help us to understand how unnatural it would be for RNA to replicate itself. Fact number one, RNA is a string of nucleotides, individual letters in a string. It's a polymer. And each nucleotide has 16 different chiral forms. You've often heard, I'm sure, that this chirality problem is a big challenge for the origin of life. And often people speak of chirality as left-handed and right-handed. But when it comes to ribonucleotides, you actually have 16 different chiral forms for each ribonucleotide. So to put that in perspective, if you want an RNA that's just six letters long, a very, very short RNA, you're already up to over 16 million stereoisomers are possible. And only one of those is used for life. So somehow you got to filter out 16 million bad ones and pick the one that's good. It's quite a challenge. And then compounding that is that each of the pieces inside a ribonucleotide can join together in different ways. You got the base, the sugar, the phosphate. They can join together in different ways. And then the ribonucleotides can join together in various ways. And life just has one canonical form. You always have to have the 3', 5' phosphodiester bond between each of the ribonucleotides in life. And that's not so easy. It's like creating a lot more isoforms that are not useful. And then the ribonucleotides have to be put into a high energy state in order to overcome the thermodynamics. They don't naturally want to bond to each other in water. In fact, it's quite the opposite. They want to separate from each other in water. So to overcome those thermodynamics, you got to put it into a high energy state. Usually it's the triphosphate state. And that's rather unnatural. I mean, I'm not saying it can't be done, but it's a special condition that is unstable also. It doesn't last very long when you're in that high energy state. And then the RNA must be folded up into a shape in order to function, to do its job of catalysis. But in order to be duplicated, if you're going to duplicate it, it has to be unfolded. So how can you get a molecule to replicate itself, if it has to be folded in order to work and unfolded in order to be read and replicated? That's a paradox. And then, in order to replicate it, you must first create its complementary strand, kind of the negative or the inverse of it. And the strand and its complement really stick together. And the longer they are, the more they stick together. When you get to about 30 nucleotides or longer, which is a relatively short RNA, you can't even separate them by boiling water. And the high temperatures, of course, lead them to degrade very rapidly. So that's a big challenge. We also know that compared to DNA, RNA is much more fragile. There's a lot of situations that just tear it apart, including higher temperatures. And then if you could get to a place where you can actually replicate lots of RNAs, there's quite a challenge here in that you would tend to replicate the smaller and smaller RNAs because they replicate quicker, easier, and you end up with a pool of smaller RNAs with decreasing information content. And these are what we call parasitic RNAs. It's not advancing toward complexity as you would hope. You're actually advancing toward simplicity when you do this. And number 8, very high accuracy is required, or else you have this information catastrophe or error catastrophe. You lose all your information that you've built up. So that just gives you a feel. There's some other ones I haven't mentioned, but it's quite unnatural, quite going against the current, trying to swim upstream here if you think you're going to self-replicate RNA.
If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together, and I will share with you about why I embrace the resurrection of Jesus.
So back to our big ultimate goal is to find a natural explanation for the origin of life, with the sub-goal we're focusing on of replicating RNA, self-replication of RNA. Now we're actually going to take it a little smaller to the sub-goal, where we can just, for now, push aside all the concerns over, is this prebiotically plausible? Forget about the chirality, forget about the purity, and all the other stuff that could get in the way. We're just, these investigators, they simply want to show proof of concept. Even with all the human intelligence, all the perfection, all the purity and concentration and cleanliness of their conditions, they just want to show proof of concept. And James Atwater, Philip Hollinger are established leaders in this area. I think Gerald Joyce is another one. But today we're going to focus on these guys because they've recently produced a couple of papers that are of interest. That's our focus. So we want to know, how close are they to demonstrating self-replication of RNA? And their two champions, these molecules that they've developed, are right here. This one is quite a complicated RNA. You can see the strand of letters that make up this RNA. It's actually 2 parts. It's a heterodimer. And together it's about 288 nucleotides long. Quite complicated. And everybody admits, even the authors admits, that this is far too much complexity, that natural random processes on their own could never have generated such a thing, given all the history of the universe and all the mass of the universe. This is just way too complicated. And we're going to talk about this one first. I'll pop up the references here. These are recent papers that they've written. And then a little bit later, we're going to talk about this simplified ribozyme that they've come up with. It's just 45 nucleotides, and it's able to copy RNA. So first we talk about this complicated heterodimer ribozyme. So this paper came out just a few months ago, and it's quite a complicated title here. But the goal that they're trying to show here is to solve these two problems, two of the problems I've mentioned. That RNA must first be folded in order to function, but has to be unfolded in order to be copied, in order to be a template for copying. And the second problem is that complementary strands of RNA have this bad habit of sticking together and very difficult to separate. So they did some very innovative work here and came up with a specific way to solve these two issues under very specific conditions. They found that if you have acidic conditions, pH of 3.6, and you have a temperature of about 80 degrees Celsius, so we're getting close to boiling, and you have to get magnesium and other divalent cations out of the picture because those really destroy RNA. And then you add pure trinucleotides. So you're not adding single letters of RNA, but it's a triple. So it could be like AAU, something like that. And you add those triples. In this situation, those triples can kind of invade the longer RNA and unfold it, or even take apart the complementary strands. Because there's some jiggling naturally going on, especially at high temperatures, and you can get your little trimer, we'll call it a triple, these trinucleotides, to enter into an opening that temporarily opens up. And it kind of is a ratcheting effect to peel apart the two strands. Then you have to rapidly cool it to minus 7 degrees Celsius, and they do liquid nitrogen in order to cool it rapidly. You got to cool it rapidly because those high temperatures are really damaging to RNA. So get it down cold quickly. And by cooling it like this, you get almost all of the water to form into ice crystals. And that concentrates the RNA and the trimer, the triplets. It concentrates the things that you want interacting in the eutectic phase between ice crystals. And then you have to actually increase the magnesium content, because the ribozyme, which is the catalyst here, needs the magnesium. The magnesium is helping the ribozyme to form these covalent bonds. And in that situation, because it's so cold, the RNA moves very slowly. So we're talking about weeks. You have to leave it sit for weeks at this cold temperature in order for these bonds to form with the ribozyme. And then they found that you can do cycles of the above. So you heat it up with acid, you cool it down and make it more basic, and you can do cycles if you have lots of human intervention there to clean up the waste and keep homeostasis, keep everything balanced. So if you do it just right, you can actually end up with positive output, positive production of the things you're trying to make. So in other words, you can make more than what degrades.
So Rob, you're being very kind to them. These are extremely specific conditions. You go from a high temperature of 80 degrees, then you cool it down to minus 7 degrees. That in itself would be hard enough to somehow manage on anything like an early Earth. So these people certainly push this toward an origin of life type scenario. This is beautiful work, but it has nothing to do with what might have happened on an early Earth. Then, concomitant with that temperature change, you have to change the pH from 3.6, which is acidic, to pH 9, which is basic. And you have to have those oscillate as well based on the particular temperature. Now, magnesium degrades RNA quite rapidly. So they have to keep it at a very low concentration, but they need some in order to get this chemistry to work. But then there's all these other reagents that they put in there that have nothing to do with an early Earth, that are these prescribed conditions that would be hard to see how they could ever use. So for example, they use a surfactant called tween 20. Tween 20 is based off of a sorbitol, a sugar, Now, we already know from more recent work from Krishnamurti that the foremost reaction does not make these 5-carbon or 6-carbon aldoses. Where we got sorbitol, we don't know. But you take sorbitol and you put on multiple ethylene glycol units, and then at the end of one of those, you put on a lipophilic chain. That's one of the reagents that they add. Another one that they add is a CHES, C-H-E-S. This is a cyclohexylamine moiety that then has an ethylene oxide that is then sulfonated. So it's a multiple-step process to make this organic compound, which they have to have in there to stabilize the system. If they don't have that, it falls apart. The other thing they have to have in there is they have to have a special kind of buffer, which is a mixture of formamide, which is another organic compound, and then they mixed with that EDTA, which is this chelating compound. Again, all of these are human-made, human-designed groups. This ethylene diamine tetraacetate. This is, again, very hard to think of how this would ever come on an early Earth. It's all a synthetic compound. And then they have bromophenol, another organic compound in there. And so they have all of these different reagents. They also have tris, this tris hydroxymethyl aminomethane. It's again a compound that has to be prepared from formaldehyde and ammonia, and so all of these compounds have to be in there. So you're being really kind because you're not showing all the other support materials that are needed for this. So this is a highly sophisticated biochemistry experiment, but it has nothing to do with life's origin. So their implications of this, I think, are not well suited. And to start some of their papers talking about the origin of life and this self-replication, it's unfair because it throws off the public. Anyway, that's my thoughts on this. Go ahead.
Yeah, very, very good to add all of that. And of course, they just purchase all that stuff that you mentioned. If they had to actually generate it themselves, that would take a factory and lots of years to do that. And I also can add to what you just said that when you do this cycling of high temperature, low pH, and then low temperature, high pH, you build up ion. You keep adding stuff to do the cycle, and you end up with a lot of extra ions in there. And they actually have to flush it out and clean it up every now and then. Every fourth cycle, I think they had to clean it up. So there's a whole lot of human intervention the whole way through this, and we can't lose track of that. And then this will kind of add a little flair to what Jim just said. These are the trinucleotide triphosphates that are the basic building blocks they're using. So they're not using single monomers. It's a triple here. And you can see it's quite a complicated molecule. This is a hard thing to even go out and buy because it's very complicated. And they come in 64 varieties because this is the AAA variety, right? And there's 4 different kinds of each one. So you can have 4 times 4 times 4 is 64 varieties. And as I mentioned, the chirality is quite a challenge because you could have 4,096 different stereoisomers of this compound. But they went out and bought them, and they bought them with, of course, just one chirality.
But yeah, only one stereoisomer works of the 4,000, and you buy it, and each one of these, you've got 3 units there, but each one is a combination of 3. So you have a sugar, and then you have a nucleobase, and you have a phosphate on each one. So it's actually a trimer of trimers in that sense. And all of these are gotten from natural sources. So you take cellular material and you get these from natural sources, and then you go into a laboratory, a commercial synthetic laboratory will take these from natural sources, and then they have to run off of beads or gels, beads to synthesize this into a trimer, and then cap it off with the triphosphate at the top. So that again is not available on an early Earth, not even close. And just the sugar portion we know cannot form on its own. I mean, the foremost reaction doesn't do it, doesn't make the 5-carbon or the 6-carbon sugars, according to recent work that was published in 2025. So again, has nothing to do with life's origin.
And to clarify what you said they come from natural sources. You're talking about things that are borrowed from life, from
living
things.
living
Yes.
cells. Yeah.
Not natural in the sense of like a prebiotic world with just molecules bumping into each other.
Correct.
Right?
That's what I meant.
Okay, so this paper that did this cycling of hot and cold and showed replication of lots of different kinds of RNA, not replication of the ribozyme itself. Anyway, it caused a lot of hype. So as you always know, there's this hype that comes out and the media plays it up. And so here they say scientists are pretty close to replicating the first thing that ever lived. So it's interesting that they're actually even claiming that a single RNA molecule was the first thing that ever lived. I think that's pretty suspicious. And down here, chemists recreate how RNA might have reproduced for the first time.
Yeah. And what's crazy about this is RNA is so far from living, so far from life. There are a million things that have to be made in addition to RNA in order to have life. And then those million things have to assemble in ways that we can't even fathom in order to have life. But they love to throw that word around of life and living. It's no wonder why the world is so confused on this.
Totally agree, yes. So just to give a little bit of an animation of what they're doing here. So this is a strand of RNA, and usually this is going to be folded, right? There'll be some hairpin turns here, and it'll stick to itself where the nucleotides match up and pair up and stick to itself. So I'm showing you the straightened version of it. But the way to unfold it is that they used 80 degrees Celsius acidic conditions with low magnesium. So that unfolds it, and now it's there available to be copied, kind of like a template. And then they increase the pH to 9, and then they add a primer. And this is something we haven't mentioned yet, but this is kind of a little trick up their sleeve. They're kind of giving the RNA replication process a head start. So they pop in this little 9, I think this is 9 nucleotides here, and it's already preformed, and they give it for free. They
buy that, right?
Yeah, they buy this, and of course it's a perfect match here, as you can see.
Each one of those nucleotides was taken from a living cell, and then they buy the manufacturer will hook this together for them in the 9 configuration using totally modern synthetic methods. Never would that be available on an early Earth.
And this is needed to give the ribozyme a bit of a head start, that it knows where to bind and to start to do its job. And then they start adding these triples like this. So they could throw in maybe all 64 flavors of the triples, but they bind up here because these are matching, complementary, and they match up like this. And sometimes actually they found that they don't match up so well, and they don't stick so well. So sometimes they have to put in a hexamer. So that's a 6-fold. And I told you this hexamer has over 16 million different stereoisomers, and only one, of course, is what they put in there because they've already they bought the one that they needed, and that's what's available here. And so the process goes on, and so now you've got this nice pairing process going on, but you can see there's no covalent bonds yet between the different parts that they've added here. So they got to bring in their workhorse, the ribozyme. But for that to work, they have to increase the magnesium and rapidly cool it to -7. And so we bring in the ribozyme, which I picture here as just a blob. But that blob is actually also a sequence of RNA nucleotides like this, which is doing the bonding. I mean, that's that big heterodimer molecule that I showed a couple slides ago. And so that comes in here, and it's going to form this covalent bond. But that covalent bond also has a head start because it has that triphosphate. It's in a high energy state, and it's more possible than to overcome the unfavorable thermodynamics of this reaction. So as it comes along and makes these bonds, and I'm showing this in very high speed because what's going on here would actually take days or weeks in their reaction. But each time it tries to make this covalent bond, they say there's about a 20% chance of failing to complete. So right here, you see it failed to complete that ligation. And there's also a fidelity challenge here, that about 94% fidelity to match each nucleotide. Those who are really paying close attention could see that this right here is a mismatch. A should be matched to U, but we have a G here, so that's an error. And their hope is that that kind of error might produce a more functional molecule, a better molecule, and advance toward life. But it also much more likely creates something that's not so functional and causes trouble. And to compare it to existing life, the least accurate DNA copying process in all known life, this is in Mycoplasma genitalium, very, very simple kind of bacteria. It's over 1 million times more accurate than this RNA copying process we see here with 94% fidelity. So there's a long way to go. So now we have created, you'd say, the complement to the RNA strand. This is like the plus strand. This would be the negative minus strand. Of course, it's not really a complement because there's a problem here. There's no covalent bond, but let's give them the benefit of the doubt. So the next step would then be to separate these two from each other and then use that minus strand as a new template to try to make the plus strand again, and that is replication. So you finally have made a copy of the RNA you intended to copy. So that's kind of how it works. And now we're going to move on to their latest work. And this is actually not published yet. It's in a pre-publication format that you can pull up on bioRxiv. And I like the title they picked here, a polymerase ribozyme that can synthesize both itself and its complementary strand. I'm afraid a lot of people will read this and say that we now have self-replicating RNA.
Yeah. When you just read the title, it's going to throw you off. Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved. That's right. That's the requirement. We talk about science concepts which draw people in. Take these nanomachines and have them drill into cells. This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything. My faith in Jesus Christ means more to me than anything. If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandscience.org/donate. All U.S. donations are tax deductible. Thank you so much.
I think they're trying to be modest about it, honestly. Maybe they could have tried a little harder, but it doesn't synthesize itself. As we'll see, it can separately, under perfect conditions, synthesize its negative strand, and then from the negative strand can make a positive strand in a separate reaction under different conditions. That's the challenge here. So they're working now with a smaller RNA. It's only 45 nucleotides. So that's a nice advancement, really. They spent 10 years working on the previous one. And Phil Hollinger said it took a lot of courage kind of to give up on 10 years of work on that one and start fresh. So with a lot of human intelligence and an artificial selection process, they've arrived at this new ribozyme, and they want to see how well that works. And this is the paper where they had that statement in the introduction that I really want to emphasize. They say that the ability for RNA self-replication has not been observed in nature so far. So in this paper, they're focusing on this particular paradox of it needs to be folded up in order to be functional, but needs to be unfolded in order to be copied. That's quite a challenge. And their approach, pretty ingenious, is to try to sidestep this paradox by providing just the precise conditions that would be needed to have equilibrium between this ribozyme being in a folded state and an unfolded state. So if you provide just the right conditions as this ribozyme is jiggling and moving around, some of it will be open, some of them closed, open and closed. And so you can get this delicate balance, trying to copy it while it's open. Okay, so we're going to focus on what exactly did it take in order to have this thing make copies of strands. So first we're going to make the complement. So the ribozyme itself is the positive strand. We're going to make the negative complement. And the first trick they do is that they extend the ribozyme by adding 10 nucleotides to the 3' end. So in other words, they took down here the 3' end and they did a reaction in the lab there to add 10 nucleotides onto it. And the reason why they do that is they're going to match that up with the primer. We talked about the primer, and they want to have this extension and the primer, leaving this 45 nucleotide RNA by itself so that they can claim that it copied this strand. But it can only copy the strand if you add the 10 and the primer onto it. So it's a little bit of a convenient cheat, I guess. And you have to put the exact amount of this template, this extended template, in this concentration, and the right amount of primer that matches same concentration of the primer. So then the primer binds to this extension. And then you have to have the right amount of the ribozyme added. And then very carefully, this is a really important number, the right concentration of each of those triplets, those triphosphate trinucleotides, 64 flavors of them, and the right amount of magnesium and the right pH and all the other things that Jim mentioned that are part of the buffering. And you leave it for 28 days at -7 degrees. And with all that in mind and all that human intelligence and intervention, they were able to show that they could make the complement, the negative strand. So that's kind of like a big success on, they're going to claim success for that. But now they have to take the negative strand and make a positive strand. And you would think that they could just take the negative strand that they produced.
Wait, Rob, what was their yield of the negative?
Well, that's on the next slide. And they hinted that it's like in the 1% kind of range, but the paper did not quantify that.
My experience with scientific papers when they're dealing with chemical questions is that if they do not list the yield, it's a terrible yield. If it's a good yield, it'll always be listed. But if they hinted it's at 1%, that means you get a lot of other junk in there that would contaminate this reaction. But it's telling, and their lack of specificity on that is also quite revealing.
I totally agree. Let's look at it here. This is the making of the complementary strand we just talked about. So after 28 days of reaction time, and they run this blot, and the idea is that as the RNA complementary strand is produced, it grows longer and longer, and that's kind of like climbing up this ladder on this blot. And your goal is to get to here. This is the full-length final complementary strand. That's the desired product. But you can see here, this is not quantitative, but it gives you a qualitative feeling of all the other junk that was produced after 28 days. You produce all this stuff that's garbage that you don't want, and you produce this much of the product you do want. And that is better if you only put in the 15 kinds of trinucleotides or triplets that you need in the reaction. If you put in all 64 flavors, which includes some that you don't want, you can see that the yield goes down dramatically. So this speaks to the required purity. If this chemical system only has the stuff you need, then you're kind of pushing the yield higher. But if you're in the middle of a bunch of stuff you don't need, the yield goes down quite dramatically. And imagine if you also had millions of other compounds of stuff on a prebiotic earth, including things with the wrong chirality, the wrong bonding, mononucleotides, all kinds of junk, then the yield just dramatically drops off a cliff.
Right, and what did you say? You said there was like over 1 million different stereoisomers from that 64?
Each one of the 64 has 4,096 different stereoisomers, yeah.
Right. So yeah, it's 4,000 times 64.
Yes, and that's just for triplets, trinucleotide triplets.
That's just for the trinucleotides. So you're going to have some dinucleotides. You're going to have So this
all
is
kind of junk.
This is a real mess that has been figured out by amazingly smart human beings to try to solve this thing. And even with all their solution, their yield was about 1% or less. So that's the state of what we've got.
Yeah, and so you would think that, ideally they would start the next step, which is to make the positive strand, that they would start by just using the negative strand they just produced. But the problem is, like I said, all of this stuff down here is just garbage that gets in the way. And you can't, this very tiny amount they produced, it just isn't useful in order to move forward. So backing up here, plus I forgot to mention that the negative strand that they did produce also still has this old extender stuck to it, that they stuck to it. So it's sort of an extra burden, and it's not actually the molecule you want anymore. So what they did after they produced the negative strand and claimed success is they just threw out that entire reaction, right? And start with a nice, clean, new system. Start fresh.
So they didn't use what they made.
Right. Nor did they even speak about the fidelity of what they made. I mean, even if they got some of it that was the right length, it may not have been functional.
Okay.
It may have the wrong bases. Yeah.
Right. So they didn't speak about how clean it was in its formation. But this is what I talk about in a sense of relay synthesis. They couldn't make the template from one, make the complementary, and then make the complementary of that. They had to start, buy something from a manufacturer that's of a specific arrangement, and then do the next step. This is the whole relay synthesis problem. They've just done it here again. This is just so full of errors in the way it is that it could never be thought of to be used for life's origin.
Yep, lots and lots and lots of intervention here. It's great to keep that in mind. So now their goal is to make the plus strand, given the minus strand. And there's kind of a new challenge, because as soon as you put the minus strand in, with the plus being the ribozyme itself, those two are going to stick together. And you get a strand duplex. And when they stick together like that, it's basically game over. You just have a pile of junk that's not functional anymore. And so in the paper, there's a quote, "The strand inhibition problem caused by the formation of the ribozyme pairing up with its negative strand is a key obstacle to closing the replication cycle." So this was a big challenge, and for a while they couldn't get it to work. And then they found one particular condition where they could kind of force it to work. So what they did was they take the negative strand and they add this extension to it again, right? Like we saw before, they add that to the 3' end. And they have to have much less template this time. Last time it was 250 nanomolar, now it's 8. So it's like 30 times more dilute. And that was required in order to get it to unfold and have the pairing happen. So, and separate the plus and minus strands. So they reduce the pH, heat it to 80 degrees rather quickly, make sure there's not much magnesium or other divalent cations. Then they found that they had to add 1 hexamer, so that's six letters of RNA. They had to add that in there so that it would quickly bind to that template and kind of block the possibility of the plus and minus strands sticking together. So it was kind of physically interfering with this re-annealing process. And then they could add 8 nanomolars of the primer, which matches up perfectly to this extension. And then they try to make it produce the plus strand. And they found that it only worked if they only provided each of the 13 required triphosphate triplets in the right concentration. It didn't work at all if they added all 64 flavors this time. So you could tell there's a lot of intervention there. So this is the result, the blot, if you only added the 13 trinucleotide triplets that were needed and not all 64. And you can see the yield here. And that's after 33 days at minus 7 degrees.
Yeah, so whatever they got, it's a very low yield.
Very low yield. And now we could talk about the fidelity of it. So in blue here is the actual ribozyme itself, and in orange would be the complement that they were trying to make, the minus strand. And now they show the accuracy of matching, of copying each one of the 45 nucleotides. And it's a little interesting because this one nucleotide, this guy right here, they found it only had a 20% chance of being accurately replicated in that one spot. And this is color-coded to be a heat map, whereas the red color indicates that it's a really critical nucleotide to get correct. So if there's an error in the more orange-red colored ones, you end up with a dysfunctional ribozyme. So this is a pretty sensitive area to have mistakes, and they had a lot of mistakes happening there. And then if you did the same thing to try to go from the negative to the positive, again, you have this kind of problem, and the accuracy sort of compounds over time. And the paper itself doesn't actually talk about how functional the actual product was. When they finally made a plus strand, did it actually have any function to it whatsoever? They didn't mention that, and that makes me suspect that it just didn't work. It was broken.
Yeah, if it had worked, they would have said it.
I would think so. So back to the top, we had this ultimate goal and a sub-goal and a sub-sub-goal, which the sub-sub-goal was just forget about all the prebiotically plausible stuff. Let's just show proof of concept, pull out all the stops and see if you can make it work. And so this is their best effort. And they are very smart guys, and they did some pretty amazing, ingenious, innovative work. But there's still some things missing from achieving this sub-sub goal, which is, as we mentioned, the yield is pretty bad. They also mentioned in the paper that this ribozyme, they had artificially evolved it to be where it's at. And they show that there's a really sharp fitness landscape to it. The quote here is, almost all mutations being detrimental to activity, meaning that it doesn't have a lot of room to get better. It's kind of already optimized, and it's not going to get much better. And they didn't really have any demonstration that the replicated RNA that they made is actually functional, and certainly they haven't gone to the point of having a cycle of replication that could produce exponential growth, which is what chemical evolution would require. And as we mentioned, it took tons of human intervention, including starting over between plus and minus. So to just wrap this up, they're claiming progress toward RNA self-replication, but the results show that we're still a long way off from the proof of concept of RNA self-replication, even with intense human intervention and completely disregarding what is prebiotically plausible. And we're a long, long way off from thinking that self-replication could occur in nature by itself. And we're a really, really, really long way off from believing that any of this could contribute to the origin of life. I just want to emphasize this last statement because this really, to me, is something to focus on, that because it took so much intervention and so much human intelligence in order to achieve what they did achieve, and of course there's all those limitations. So doesn't that really prove to us that some intelligence, a lot of intelligence, was required to get life started in the first place? I think that's what they're really proving to us.
Yeah, it's like I say all the time, Rob. The work that they do themselves screams out that life did not start this way. This is showing that life did not start this way, because even with all of their input, even with all of their human-made reagents that are only available in a modern laboratory, and all their ability to have pH control and warm and cool like this with precise pH control, with precise reagents to stabilize, only then did they get it to work in 1% yield or less with a fidelity that was not mentioned. Never was it shown to be functional, and plus it would be contaminated by all that other 99% of the material that's in there. So this screams out that life could not have started this way. People are really clever, but with all their cleverness, they're still very far from being able to solve something related to life in a laboratory. And plus they had to start with all of the basic components came from living systems. If they had to make all of these components themselves in a laboratory under prebiotic type methods, they would have been unsuccessful.
And you know, I kind of view this too, this whole video we're producing here is a bit of a forewarning, because I can predict it now. I can see in a few years, this is going to keep happening, of course. And in a few years, there will be a paper that comes out that claims self-replication, right? And it goes back to everything we've shown here, that you got to read the details and pay attention to all the human intervention and all the cheats, you know, that went into it. So they may be trying to prove proof of concept, but the practical application of it is very, very distant from what they may be claiming in the media hype and maybe even in the paper itself.
There's another indication that we are getting further away from life. We're not getting closer, we're getting further away. With that, we're going to wrap it up. Thank you, Rob, for going through that paper in great detail. You know, to go through a paper like this, to really think about what's done is not easy. And it takes a lot of time, and I thank you for putting in that time and bringing us through it. Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.